Hi, everyone, and thanks for joining our second annual CARSS webinar. My name is Sahar. I'm the director of CARSS, which stands for Center for Autonomic Recording and Stimulation Systems. I oversee the operations and technology integration of our open source implantable neuromodulation system known as OpenNerve Platform. So a little bit about who we are and how we're funded. We're funded by a sub-program within the NIH Spark initiative, and this program is dedicated to accelerating the development of neuromodulation platforms, specifically bioelectronic medicine. So through this initiative, a key issue with current clinical grade neuromodulation platforms was identified, and that is that they're either very costly to develop from scratch or too difficult to access for exploratory use. So our objective at CARS is to advance the clinical translation of bioelectronic therapies by sort of creating an open source clinical grade system. And through that, we'd like to enable interested users to either replicate the exact design or customize it to meet their specific requirements. So now the team responsible for development of each component. So we have 3 USC investigators and 2 industry partners. We have doctors Ellis Smeng, Amral Musavi, and Hengo Zhao from USC. They're focusing on development of innovative cuff electrodes and sensors. From our industry side, we have Mr. Raja Hetty from MedAlly and Dr. Victor Pikoff from Medipace. They're concentrating on development of more established components, which include electrodes, various sensors, and all the mechanical components, software, and hardware that are required for an implantable pulse generator. The 2 industry partners, they've strategically selected these components to accelerate the development process within the 3-year program timeframe that we have so that we can achieve an example FDA regulatory approval milestone for a target use case. We also have an admin, technology integration, and dissemination team here at USC, and we're actively seeking collaboration opportunities with users, assisting in submission of grant proposals, management of animal studies and data collection, and finally publicly releasing design files, codes, and data. And so we have a pretty large team, and this doesn't even cover any of our current collaborators / users that are currently working with us, which we'll likely discuss more about in the next webinar. Now I'll talk about our system, a little bit about the animal studies that are underway, and a high-level time frame of milestone. So here's a rundown of the neuromodulation components we're developing. This system includes an implantable pulse generator, or IPG, with Bluetooth for communication between patients and clinician or any other remote devices for the purpose of reprogramming stimulation parameters to modulate the nerve differently. So this comes also with a range of electrodes as well as sensors for closed loop mode or for sending alerts to patient clinician via Bluetooth. Our ultimate aim is to build each component and establish the system as an open source initiative through wiki and online repositories. We've structured our project around 5 nested elements, and they're referred to as nests. Each are categorized into 2 main types. So we have our established components that are developed under quality management standards, and our novel components that are undergoing feasibility testing. The established components are nests 1 through 3 that consist of an IPG mechanical and a comprehensive array of sensors and stimulation electrodes. This includes sacral nerve stimulation, PDMS cuff, ECG, EMG, and accelerometer leads that are all of these are developed by MedAlly. The electronics and firmware components of the IPG are developed by Medipace. On the other side, our novel components include Nest 4. Nest 4 is developing a microfabricated cuff electrode and surgical tools, and it's specifically designed for interfacing with small branch fibers of less than 1 millimeter. And this is developed by Dr. Ellis Ming's team. Nest 5 is divided into two. So we have temperature sensors for monitoring inflammation, strain sensor to detect changes in static movement, and both of these are developed by Dr. Heng Boja's group. And then lastly, we have a pH sensor, which can be used as a biomarker of disease, for example, stomach acid, and chemical sensors that can be implanted at the nerve ending with acetylcholine and or catecholamines as target chemicals. These two are neurotransmitters of parasympathetic and sympathetic autonomic nervous system. So this is a mock-up of the IPG along with various sensors. The IPG is very modular and can accommodate up to 16 contacts, which makes for a very flexible design. In the middle, there's an interconnect that can connect up to 4 contacts per lead. So you can have a 4x4 contact lead. Another feature that I hope you can appreciate is that we are using off-the-shelf ball seal connector with set screw for interchangeability with other technology. So the team's focus so far has been development and benchtop verification. We're looking forward to conducting some animal studies, first to demonstrate the safety and functionality of all components, and then some follow-up studies to demonstrate their functionality for specific use cases to sort of help with disease-specific system development. And these are all thanks to NIH for giving us the opportunity to conduct these studies through supplemental awards. So the first supplemental, the funds and the IACUC protocol are both approved, and we're just starting the in vivo studies. In this project is divided up into 2 categories. So we have Nest 1 through 3, which are components from our industry partners. They will undergo 3-month chronic PIC studies. Nest 4 and 5 are our novel components, and they will undergo 1-month rat studies. In the second supplemental, we just submitted IACUC protocols, so we may start sometimes in August. Here, we identified experts in the field working to treat specific disorders that would benefit from our OpenNerve platform. So during this work, we'll conduct short-term and long-term validation in large animals, one for bladder dysfunction, and this will require sacral anterior root stimulation. 2 for hypoglycemia and type 1 diabetes, and here we'll use abdominal vagus nerve stimulation using the Perlin cuff electrode developed by Nest 4. And 3, unidirectional nerve block with a broad range of potential application this can have for researchers who are looking to suppress action potentials traveling towards the brain or vice versa. In the last supplemental, similar to the second supplemental, we'll focus on disease-specific studies, and the options that we submitted are 1, chronic constipation through sacral nerve stimulation, and then the other one is stroke-induced dysphagia using vagus nerve stimulation. So all of these are closed loop, and we'll use various sensors from OpenNerve Platform, and then we plan to release the data and the results from the studies through Spark DRC. So stay tuned on that. So here are a few other examples that we're not currently working on. And if you have a novel indication in mind with the expertise that could utilize any of our components here, make sure to get in touch with us. And I've listed our website and our contact info at the end of this presentation. Okay, so in summary, who can potentially benefit as the intended user of OpenNerve? Essentially, anyone with bioelectronic therapy that require hardware, software, and firmware for research studies. So let's say if your idea requires components from our academic partner, the first question is whether there are surgical techniques available for your target organ. We then need to figure out any necessary modifications to make the surgical technique work and then conduct some feasibility studies. If your needs involve using established components, the question shifts towards whether there is research supporting the intended therapy. And so we discuss validation of the therapy in large animals. In both of these scenarios, you could simply grab the design files, tweak them as needed, and then manufacture them through your chosen manufacturer. Alternatively, you could collaborate with the appropriate nest and our manufacturer MedAlly through a funding mechanism, which we highly recommend because you can then utilize the team's expertise to address your needs and then provide guidance in any modifications that you may need. And if you're unsure about which path to go in any way, please feel free to contact us and we'll sort it out for you. Lastly, here's a timeline of our project. The center was launched in September 2022. So far, we've developed and released initial design files. We've also established test procedures and are currently conducting benchtop studies. In the meantime, we've identified several indications through conferences and direct contact with experts. We've also received supplemental awards to conduct animal studies for specific novel indications. And by the end of 2025, we aim to have an integrated system ready for clinical use. We hope that some of these indications we're working on will reach this stage so that we can provide example documentation on development and regulatory processes for a potential use case. So this wraps up our high-level overview of the system. I'd just like to emphasize here that an open source implantable neuromodulation system is a very novel idea with numerous challenges. We're actively trying to address these challenges and we're hoping to foster a collaborative environment to bring innovative therapies to the market faster. So please stay on to hear from each team working on specific elements of the system. Finally, we'll discuss our strategy for sharing open source content, delve into collaboration examples, and cover IP and licensing. And please be sure to visit our website for more details, reach out with any questions, and follow us on LinkedIn for updates. Thank you very much for your attention.
Hello, everyone. I'm Jamie Coates from MedAlly, and I will be sharing about Nest 1 and Nest 3. MedAlly was established in February of 2011 with a focus on active implantable medical device development from design through production. Located on the outskirts of Charleston, we are an ISO 13485 company. We have 10,000 square feet of production with 3,000 square feet of clean room. Our capabilities include laser welding, hermetic testing, robotics, braiding, molding, and more. We also provide full design and development services. We are working on Nest 1 and Nest 3 as part of this program. In Nest 1, we are focused on the mechanical enclosure of the IPG along with a charger. And in Nest 3, we are focused on a set of standard available leads for the system. For Nest 1, we are implementing technologies that will enclose the electronics that Victor in Nest 2 is developing. We have developed and prototyped a mechanical enclosure that will support the module approach that the team is implementing to enable many different uses of this device. To minimize risks for the users, we have used proven technologies in the development of our IPG. The designs have been optimized for modularity, ease of prototyping, and supply chain availability. Specifically, the IPG will have the following features: a hermetic enclosure for the electronics portion that supports housing all sensing and stimulation control, as well as housing the charging electronics and rechargeable battery. In the header portion of the IPG will be a Bluetooth antenna with 4 independent lead borers with 4 connectors each. A 2-lead configuration with 8 connectors is also available, with only a small change. A hermetic enclosure will be made primarily of a grade of titanium that will be laser welded together to form a hermetic barrier and was selected to facilitate charging without overheating the IPG during the charging once implemented. The titanium case can also be marked with any pertinent labeling and identification that is desired or required by regulatory authorities. The IPG header has 4 lead access points, each with 4 ball seal IS4 contacts or 2 8-contact leads. Additionally, there are 4 sets of set screws to secure the leads in the header and 2 locations for suturing the IPG to underlying tissue. Additionally, Nest 1 includes an external charger that accompanies an IPG. Along with Nest 1, we are also developing the partnering leads as part of Nest 3. Nest 3 incorporates a series of standard leads to enable new research. An SNS, or sacral nerve stimulation lead, with 4 contacts, a cuff lead with bipolar electrodes, an EMG sensing lead, an ECG sensing lead, and an accelerometer-based motion sensing lead. The lengths of these leads can be customized per application. Additionally, there is an interconnect that will allow a 4-contact lead from the header to split into 2 bipolar leads that will allow for further customization and integration with various sensor modular leads being developed in the additional nests. The team has been able to fully prototype the SNS, cuff, and interconnect lead, and will be working over the next few months to receive a full list of components to prototype the remaining leads, which are the accelerometer, EMG, and ECG leads. Additionally, the Nest 3 team is working on integrating the sensor modalities from the other nests into the lead bodies and proximal connectors that will enable the novel sensor designs to be seamlessly integrated with the IPG. The next step for Nest 1 and 3 is to secure the supply chain and materials for testing builds, and then initiate a subset of V&V-like tests to confirm that the system meets some of the basic requirements for IPG and lead design and manufacturing for future submission to the FDA or other regulatory bodies. Thanks for the time today, and feel free to contact me at any time if you have any questions at all.
Hello, my name is Victor Pikov, and I'm from Medipace Inc. Today I will tell you about the program made in Nest 2 for development of printed circuit board assembly, or PCBA, and firmware. The IPG electronics includes 3 main blocks. The first one is stimulation, second is sensing, and the third one is housekeeping items. For stimulation, we have 3 main functionalities. One is sacral stimulation, another one is vagal nerve stimulation, and the third new one we just recently added is the vagal block using pseudosine waves. Sensing block is the largest one. We have a variety of sensors, including EKG, electrocardiogram, EMG, and EGG standing for electromyogram and electrogastrogram. Gastrogram is a type of EMG for intestinal activity. For EKG, by the way, we have 2 types of recording. One is to record respiratory rate, and another is heart rate. These numbers here represent the filter settings for low pass and high pass filtering. So together, they represent band pass filter. And of course, there is a different gain for these channels as well. In addition, have sensing from for electrical impedance from the stimulation electrodes, the ones here on the left. And finally, we have a range of additional electrons, which will not be on the IPG itself, rather it will be on separate boards, and it will be connected using the I2C interface on the header. And these include the accelerometer board, capacitive strain board, resistive temperature board, an electrochemical board that can measure microamperometry and voltammetry. Finally, we have housekeeping functions such as wireless power transfer, receiving coil management, management of primary and secondary batteries, power management, management of MCU microcontroller, and BLE, the Bluetooth low energy. And then management of two sensors on the IPG. One is a thermistor and another one is magnet. Magnet is for controlling the device. These dots represent the context on the header. So as you can, we have quite a few of these dots here. So this represents somewhat of a dilemma. How do we route all of these different stimulation channels and recording channels to the header? On the next slide, I'm again showing these different stimulation blocks, recording blocks. So as you've heard from previous talks, we have 4 connectors, inline connectors on the header. So we have to somehow route all of this to these 4 connectors. Here, an example solution, maybe not the only one, but one that kind of makes sense, where we can down select from either SNS or VNS NB channels to connector 1. To do that, we would use 0 ohm resistors in the layout of PCBA, and then in the final fabrication step, would select to populate either this 0 ohm resistor or this one, and depending on which one select, the final IPG would be able to produce either SNS or VNS station. Even though the blocks are present for both of these, the circuit boards, but the actual connection will only be for one of these 2 options. For EMG and EGG, our idea is we have 2 channels, and again, based on the filter settings, we select either EMG or EGG. And since it's only using 2 contacts, we can actually have 2 keys using the same contact, and then it will split using the kind of splitter that was described earlier to connect 2 of these leads to a single connector. EKG will have its own connector because it's quite important for a lot of autonomic applications, because we can detect both the heart rate and respiratory rate, and the heart rate can be used for deciphering information about sympathetic and parasympathetic activity, which is key for tuning the therapy. And finally, I mentioned earlier, we have the I2C connection in the header. And I think that deserves its own connector. And this can be daisy chained, so actually multiple sensors connected to a single connector. This is a digital protocol. So the sensor processing will happen on the external boards where there will be A to D conversion happening. So talk about the current state of development. We are almost completed with Gen 1 device, which will be used in our initial animal studies. We call this supplement 1 type animal studies, because these are already funded by the supplemental funding from the NIH Spark program. So these boards will be fabricated very soon, and they include already I2C management, and also will include the external board for accelerometer. We'll have 2 AFEs for EMG/EGG, and 2 more for EKG. We have a total of 4. It has a wireless power transfer management and magnet detection. The next generation will come a little bit later, just 3 months later. And this will have some additional boards, such as a capacitive strain board. Again, it's external board plugged into the header. And on the board, on the IPG board, we will have the sine wave generator for vagal nerve block. In order for this sine wave generator to properly function, it has to be synchronized with the vagal nerve stimulation channel. And in order to do that, we actually need to increase the number of GPIO pins we have, because we're running out of those on MCU. So we'll have an IO expander added, and we'll enable the circuitry for syncing these two channels in order to have a proper unidirectional block. So while VNS is applying stimulus bidirectionally, the VNB cuff will be able to block propagation of that signal in one of the two directions. In how VNS and VNB cuffs are placed relative to each other on the nerve. And finally, Gen 2 device will be ready at the end of 2024. This will have additional features to make it IDE ready, including a resistive temperature board and electrochemistry board capabilities, which again will be external board. And of course, we will also the bugs that will be determined in Gen 1 and Gen 1.5 devices. This is a drawing of the existing Gen 1.0 board. As you can see, it has the main board, which has a lot of circuits, including the MCU. There is also Bluetooth. There is stimulation channel circuits and recording channel circuits. In addition, there is battery management, and there is also a flexible board connecting it with another small board specifically for connecting to the battery. And we support types of batteries, primary and secondary. So there is two different holes here, sets of holes for different batteries. And this particular board will be at a 90-degree relative to the main board in order to interface with the battery. Finally, there is another board here on the top for interfacing with the feed throws that in turn will be facing outward toward the header. So again, this board has to be turned 90 degrees. That's why we have these flex PCB islands here to connect it. And this will be rotated 90 to connect with feed throws and also with the Bluetooth antenna and the ground pin, which will connect to the case, the IPG case. As you can see, there is also a lot of other components here, including these ones on top, which are basically 2mm connectors, standard 2mm connectors that are typically used in animal research. And this is in order to test this IPG with animals, which we'll do later this year. And this will allow to use this PCBA without actually putting it inside the enclosure. You can use it as is and just plug in different leads for either sense or stimulation, and without having to use any ball seal and connectors and any leads. And it also has the USB power supply, so there is no need to actually connect the implant battery. It can be powered using regular USB power. And in addition, it has a lot of other connectors for troubleshooting and communicating with MCU and Bluetooth. This is the same board shown at an angle to appreciate the about firmware. This is just a diagram showing different firmware components that we're developing based on different states of the IPG. So you can see there is a lot of IPG states, starting from sleep. And once the device is woken up, it goes into active or standby mode. And in active states, it has different modes such as communication, using BLE therapy mode, impedance testing, battery. We do static analysis and unit testing as well to more thorough analysis. And finally, this is just showing our developed Gen 1 firmware. The design of firmware will be completed in June and verification testing completed by the end of this year. And this just lists some of the very specific tasks for the firmware. and for MCU as well as for Bluetooth and also for controller. So thank you very much for your attention.
Hello everyone, my name is Brianna Thielen, and this portion of the webinar is on Nest 4, which is the microfabricated cuff electrode for interfacing with submillimeter nerve fibers. The Nest 4 team consists of 2 postdocs and 1 PhD student operating out of Dr. Ellis Meng's Biomedical Microsystems Lab in the Biomedical Engineering Department at USC. This portion of the project was motivated by the lack of options for interfacing with nerves less than or equal to 1 millimeter in diameter. We are primarily focused on interfacing with the parasympathetic nerves, specifically branches of the vagus nerve, as these are more applicable to autonomic disorders. Existing clinical cuff electrodes are built from bulk metal or metal foil embedded in thick silicone rubber and are therefore difficult to scale down to a submillimeter size. Additionally, stiffer bulk metals and thicker materials do not interface well with small, delicate nerves due to their difference in mechanical properties. For these reasons, we developed this microfabricated cuff electrode. The cuff and cable portions are built from thin film Parylene C and thin film metal. The cuff is then wrapped around the nerve in a spiral configuration with the exposed electrodes on the inner surface of the cuff. The cuff is sized to be either 0.5 or 0.7 millimeters in diameter, but because of the flexible nature of the thin film Parylene, it can be expanded, fit around the nerve, and will gently self-size to the diameter of the nerve. The Parylene cuff and cable are connected to a standard OpenNerve lead, which connects the electrodes on the inside of the device to the IPG. The lead, cable, and attachment portion of the device are overmolded in silicone rubber for strength and insulation. We designed 2 versions of the expandable Parylene cuff. The full cuff, shown here, has Parylene insulation between the electrodes, resulting in a mechanically robust cuff. We also have a helical design, shown here, which has 2 helical electrodes with no insulation between them except for 1 small bridge. This design is much more flexible and likely better for more tortuous nerves. We have chosen to evaluate both of these options to allow for more user flexibility. Zooming in, the Parylene cable and cuff are microfabricated in a planar configuration, shown in the top left image, then thermoformed to form the self-sizing cuff in the bottom left image. The electrodes, which are on the inner surface of the cuff, have a minimum geometric surface area of 1.5 millimeters squared, which was set based on the properties of thin film platinum and common vagus nerve stimulation parameters, using a safety factor of 1.5. The image on the right shows a zoomed in portion of one electrode. Rather than using one large rectangular electrode, we chose to use this aggregate electrode configuration, which consists of an array of shorted microelectrodes that act as a single larger electrode area. The dark gray shows the exposed microelectrodes, the light gray shows the Parylene backbone, and the medium gray shows areas of metal that are insulated by Parylene. This design was selected to prevent delamination and cracking of the thin film metal, which often occurs during chronic stimulation of large exposed thin film metals. The small areas of Parylene to Parylene contact in between microelectrodes, which appear as small light gray dots, help mechanically hold the thin film metal in place to prevent these failures. The photos here show fabricated and packaged devices in the full and helical configurations, with zoomed-in photos on the bottom showing the expandable cuff and exposed aggregate electrodes on the inner surface of the cuff. At this stage, we have built a number of these devices and run a battery of electrochemical tests to fully characterize properties such as the surface area, charge storage capacity, impedance, and charge injection capacity. For the stimulation use case, the charge injection capacity, or CIC, is a crucial indicator of device performance in vivo, as it tells us the maximum current we can safely apply during stimulation without damaging the electrodes. The CIC is measured using voltage transient measurements, in which a current pulse is injected and the resulting voltage response of the electrode is measured. This is represented in the graph in the top right corner, where we can see the input square current pulse in red and the resulting voltage in black for different electrode surfaces. In other terms, this test looks at the polarization of the electrode system in response to charge injection. When using bare platinum electrodes, the CIC is roughly 90 microcoulombs per centimeter squared, allowing a maximum current amplitude of just over 3 milliamps with a 500 microsecond pulse width. We also investigated electrode coatings to increase the CIC and allowable current injection. So far, we have tested both PDOT and iridium oxide, both of which provide a 20 to 30 times increase in CIC compared to platinum. We've only tested current injection of up to 30 milliamps due to limitations of our test equipment, but we anticipate maximum allowable currents of approximately 80 and 100 milliamps for these coatings based on the linear relationship between CIC and the interphase potential in the voltage transient measurement. These coatings can be seen under optical microscope, as shown in the images across the bottom. The dotted yellow line is encircling an exposed microelectrode, which appears darker in the right two images after coatings have been applied. We are also in the process of testing platinum iridium coatings, but do not yet have that data available. We also developed a protocol and tool for surgical placement of the cuff. The cuff is held on a placement tool, which holds it in the open configuration. It is then placed on top of the nerve and slid from the tool onto the nerve. As described previously, the properties of the Parylene make the cuff electrode self-size and softly close around the nerve. Using this protocol and tool, we have achieved successful placement on the benchtop on a nerve model, shown in the images on the left, and in vivo in a rat sciatic nerve, shown in the bottom right image. At this point, we are in the process of fabricating parts for further in vivo studies and benchtop accelerated lifetime testing, as well as doing further evaluation of electrode coatings. An SEM image of the platinum iridium coating, as I mentioned earlier, is shown on the right. After completing each set of tests, we will be releasing test results and documentation for each of these devices. Thank you for your time, and please feel free to direct any questions to the chat.
I'm Mara Al-Musavi. I'm excited to share our work related to NIST 5. Specifically electrochemical sensing aspect of the center project. We have the overall goal to be able to have closed loop neuromodulation and be informed about organ states. In addition to electrical stimulation and electrical recordings, we also want to have a better sense of what's going on chemically within different organs. We're actually interested to implant these sensors somewhere depending on the clinical indication. And you see that the input from these chemical sensors will be provided back to the IPG, and depending on the actual application that's going to be pursued in that study, then that input can be used for changing stimulation parameters, or it may be just used as information to understand what's going on with that organ. Specifically, we're looking at catecholamines. We're interested in dopamine and serotonin. We're also looking at acetylcholine, also looking at pH. So specifically, we have two different modules. We have the catechol sensing module and then the pH and acetylcholine sensing module. These two are separated based on the two different electrochemical techniques that we use for monitoring each neurotransmitter level. So the sensors are thin film. The material that's used across these electrodes are Parylene. So Parylene is resilient to moisture and oxygen permeation, so it does provide a really robust material, and it's also biocompatible. So all of these electrodes are thin film Parylene, depending on the module, whether this is acetylcholine pH or catecholamine. The thickness may be either 25 micron or closer to 100 micron thick. So you notice there are some wires coming out. So we have the lead portion of the sensor. And these that's a bit thicker, is strengthened using other materials. And then you see that there's some wires going out of these sensors. So that will be then connected to the IPG as you see in this picture. So the electrode that you would receive, should this be a study that you're interested in, would be the whole electrode along with those pre-connected leads that will then be connected to the appropriate IPG. Each electrode has an active sensing zone that needs to be in contact with the tissue. So I'll talk about the size of these sensing zones, but this contains your electrode. So this is a generic design for what a sensor could look like, and we're customizing the designs based on the actual clinical indication, as well as the type of sensors that we're building. And then we have the leads that are insulated, so only the electrodes are actually electrically connected to the tissue for electrochemical sensing. And these leads take the signal back and forth to where we actually connect them to wires to the IPG. So overall dimension for the sensor had been around 2 centimeters by 1 centimeter, and the active zone has a smaller area. which the active zone actually has to be in full contact with the tissue. All these electrodes are highly flexible. You can bend and twist them, and we can also incorporate suturing zones for securing these to the tissue. Now, this is a broad size. For certain applications, we may be able to customize this size for a specific application. Right. So we'll first be talking about the catecholamine sensing module. So we're using electrochemical oxidation of catecholamines. There is no antibody or other capture probes on these electrodes. We're using specifically the electrochemical properties of the catecholamines themselves. So we apply a potential, and that potential causes oxidation of the catecholamines. For catecholamine electrodes, we're working with thin film parylene, but the leads are all are platinum, so about 200 nanometers of platinum. The surface is then modified using different coatings. I'll talk more about what those coatings are. This is a 3-electrode cell. We have a reference electrode, we have the working electrode, and we have a counter electrode. We started by design of the sensor, and we initially worked on optimizing how we modify the surface and the position conditions. We're right now working on benchtop validation, aging studies, and we'll shortly move to chronic studies. All right. So the objective of surface modification are to increase sensitivity. We want to increase sensitivity to be able to measure these neurotransmitters at a lower concentration. We also want to enhance the electrode properties, long-term stability, minimize drift, and again, be able to increase the biocompatibility of the electrodes. So to modify the surface for the catecholamine electrodes, because this is a voltammetric system, we want to maximize surface area. So the first step, we've actually optimized to do a deposition, electrode deposition of metal nanoparticles. So as you notice that as we do electrode deposition of metal nanoparticles, we can increase the peak for catecholamines. So post modifying the surface with nanoparticles, which enhances the surface area initially, we modify the surface again using electropolymerization. So we polymerize E dot at the surface. We're creating a coating of P dot PSS. And you're modifying, you're looking at the data that's collected during the polymerization. And this mixture that is polymerized at the surface also contains graphene oxide nanoparticles. So a combination of the metal nanoparticles, electropolymerized polymer, and along with the graphene oxide creates a high surface area, a porous structure that then enhances the signal for measuring catecholamines. Specifically, the technique that we use for measuring catecholamines is called square wave voltammetry. We've optimized the parameters for the readout, but because we're looking at the peripheral nervous system and tissue dynamics, we really are not after millisecond temporal resolution. We're looking at slower changes and slower dynamics. So the temporal resolution for each scan is about 30 seconds. So typically we study selectivity, which is again determined by the position, by the chemical properties of these neurotransmitters themselves, but also how we functionalize and control the surface charge of the electrodes. So we've done selectivity studies in cerebrospinal fluid. You're noticing that typical interference that comes from ascorbic acid is not an issue for this study, and we can separate these peaks, for example, dopamine, serotonin, along with ascorbic acid that's present in most tissues. So far, we've finished our optimization and surface modification. We've done a lot of benchtop studies. We've done studies in biological fluids, and we're now in the middle of our benchtop aging studies to see how these electrodes last over time. So the second module I want to talk about is our acetylcholine and pH sensing. So in this system, we're not actually doing any oxidation or reduction. This is still electrochemical measurement, but it relies on equilibrium open circuit potential measurements. We're not really pushing current. We're using distribution of pH or acetylcholine across a sensing membrane to generate a surface charge. So we have active sensing membranes. We have one membrane for pH, which is an iridium oxide membrane, and we have another membrane for acetylcholine that is a PVC-based sensing membrane that contains an ionophore, organic ionophore that binds with acetylcholine. These electrodes follow the Nernst equation. Our fabrication for these electrochemical sensors is slightly different. We've moved towards carbon electrodes. These systems are fully laser engraved. We actually carbonize the material to form primarily graphitic carbon with high conductivity. So this is all engraved using a laser. The entire electrode has a thickness of about 100 micron. I want to show some performance from these electrodes. Like this is the cyclic voltammogram of ferrocyanide. You see that we have really nice peak separation. You're looking at a SEM of these systems. We have a high surface area, which then supports a stable signal. We modified the surface properties of these electrodes using airflow. So the carbonization is happening either in air or in argon, and that assists for forming a more stable interface for these specifically potentiometric systems. We're after a hydrophobic surface, so we generally do argon engraved for creating potentiometric sensors. The way we do the testing is modifying the lasing conditions to achieve the most conductivity and the best mechanical properties. And then we test further performance for the sensors. So a typical response that we get from these sensing modules is potential, electromotive force or EMF versus log activity or concentration of your target ion, and you would have a linear correlation in this plot. So we look at the sensitivity, which is then defined by the Nernst equation, whether this is behaving as theoretically expected or not. And then we look at stability, measurement range, etc. And same for the pH sensor. We're looking at hysteresis, slope, and stability of the film over time. So far, we've optimized the engraving conditions and the position conditions and the composition of our sensing membrane. We're right now in the middle of our aging studies and long-term performance studies, and then we'll shortly move for acute and chronic studies in the rats. So summary, we're moving ahead as planned based on the milestone set for the work, and I'm happy to take any questions.
Hello everyone, my name is Xing Hao. Welcome to this webinar. I'm a PhD student from Nest 5 working with Professor Hangbo Zhao and Dr. Xuan Li. In this session, I will show you the temperature and strain sensors used in our CARSS project. Let's look at our temperature sensor first. Implantable temperature sensors are invaluable for monitoring the functional status of organs, detecting symptoms of disease, and assisting in therapeutic interventions. Many kinds of infection and inflammation can lead to organ temperature change. The two research studies on the right side show temperature sensors on organ surface can provide useful information for organ transplant rejection or intestinal inflammation. Traditional thermometers, often bulky and rigid, are unsuitable for these applications. Flexible and soft temperature sensors are usually required for modern therapies. Here, we aim to develop a thin film temperature sensor with 0.2 Celsius accuracy and has high repeatability and stability. The mechanism of our temperature sensor is the resistance change effect of metal thin film when temperature changes. Briefly speaking, resistance of temperature sensor varies linearly with temperature. Here, we use the small line width, thin layer, and serpentine structure to amplify this temperature sensing effect, thereby increasing accuracy and flexibility. Using classic lithography-based method, we fabricated these temperature sensors. The main line widths are 10 micron and 20 micron, and the resistance are 30 and 100 kilo ohms. Our sensors were calibrated using the gold standard, which is mercury thermometer in a precision measurement setup. The data shows our sensors have pretty good repeatability and linearity. In addition, the accuracy is within 0.2 Celsius and the resolution is 0.1 Celsius. So high accuracy temperature sensing was already realized. The current issue being addressed is the unsatisfactory long-term stability of the temperature sensor. When immersed in PBS, the testing data will be poor over time. Limited long-term stability is likely due to insufficient sensor encapsulation. As we can see here, obviously the color of the conductive, as you can see, the color of the conductive epoxy changed significantly, and the protective thin film lost its insulation after long period test. This observation suggests that the encapsulation fails to protect the sensor effectively from water moisture. Our sensor contains two polymer encapsulation layers, which are PI and Parylene. As we can see in our water transmission test data, Parylene has significantly better waterproof performance compared with PI. Even Parylene is only several microns. It is still better than 50 micron PI. So increasing the thickness of Parylene will be more effective for our sensor's protection. Based on this, we then change our Parylene layer from 5 micron to 20 micron, as you can see on our right-hand side. Here we have 20 micron Parylene on both sides of our sensors. Subsequently, we re-evaluated the long-term stability of the sensor. The data indicates that using a thicker layer of Parylene significantly enhances performance. Clearly, our newly designed temperature sensor equipped with this thicker Parylene coating demonstrates improved longevity. Long-term testing is still ongoing, and we will provide a comprehensive summary upon this completion. For the strain sensor, the goal is to measure organ deformations, which can detect abnormal organ functional states due to diseases. Many research works have tried to measure organ deformations in various animals using strain sensors. For example, a strain sensor attached on a rat bladder can identify abnormal bladder behavior, which can indicate if the spinal cord is injured or not. However, few studies have solved the encapsulation problem for stretchable strain sensors. Here, we are focusing on the large strain range and good encapsulation inside a liquid environment. We want to achieve 30 to 40% strain while having good encapsulation for several months. This is new and also very challenging because our strain sensor is using a unique wrinkle Parylene encapsulation. Our strain sensor is based on parallel plate capacitive sensing mechanism. When the two stretchable conductive films are stretched, the overlapping area will increase and the gap between the two films will decrease, thus increasing the capacitance between them. We previously used a commercially available capacitive strain sensor, as you can see on the bottom left. But later we developed our own sensor by using carbon nanotubes and EcoFlex elastomer, as you can see on the sensor assembly on the right-hand side. This can give us more customization, especially if we want to test it on different animals or organs. To encapsulate the sensor, we developed a stretchable Parylene layer. As you can see on the left-hand side, we first pre-stretch the sensor and then develop Parylene and then deposit Parylene on its surface. Eventually, we release the sensor to spontaneously form a wrinkle Parylene layer that is stretchable. As you can see in the middle, when Parylene is under compressive strain, they will buckle to form those wavy structures. We then apply epoxy and another layer of Parylene at the connector to achieve full sensor encapsulation. Such strain sensor can be stretched up to 50% strain with gauge factor larger than 2 and also minimal hysteresis. We also characterized both the sensor's encapsulation and mechanical properties in order to have a good balance between them. Here, we first measure the water vapor transmission rate through different materials. We can see on the bottom table, the EcoFlex and PDMS elastomer can have large water vapor transmission, but 2 to 3 micron Parylene can reduce it significantly. However, This thickness is still too thick. The figure on the right side shows the modulus of our sensors after forming 2 to 3 micron thick wrinkle paralleling, which is still higher than the modulus of most organ tissues. One way to solve it is to add a thin inorganic layer between two paralleling layers. The inorganic layer has much better encapsulation performance, and we can reduce the paralleling thickness to reduce the overall stiffness. For this part, we're still working on it. To verify our encapsulation performance, we have built different aging setups. One setup used a custom-made phantom organ in which we inflate it to stretch the sensor on it, but it was difficult to apply large and accurate strain on the sensor. Another test setup uses a tensile tester to stretch the sensor directly in a heated PBS cylinder. This allows us to test the sensor at a larger strain range and faster strain rate. The test is still ongoing. As you can see on the right, we are currently testing the sensor with only paralleling encapsulation. Over the span of 4 days, the encapsulated sensor showed minimal changes in its performance, and the signal was very stable, while the non-encapsulated one has very large change in its performance. To improve this, we will also apply the inorganic layer and further optimize the encapsulation for the rest of the sensor components. As for the animal test, we are currently preparing some shorter strain sensors for the rat and pig studies. In the rat study, we will suture the sensor on thigh muscle, and we will measure the leg motion as our cuff electrode triggers the sciatic nerve. And in the pig study, we will suture the sensor on the bladder surface to measure the expansion. PCAP04 capacitive board will be used to measure the signal. Since this is the chip we will eventually integrate in our IPG. Once the acute tests are successful, we will then move on to the chronic test. So thank you very much.
At this point, you all have heard a lot about the devices we're building here at CARSS, but I'd like to finish off the webinar by talking a little bit about what collaborating with CARSS might look like and how you could hopefully use CARSS resources to support your work. So just to review, we're developing an IPG as well as several simulation and sensing leads. Up to 8 of these leads can be used with the IPG, which we hope will provide the tools necessary to develop new, unique therapies. Right now, we have both clinical leads, which are based on existing leads, such as like a bipolar VNS cuff or a linear sacral nerve array, and are being designed and manufactured by MedAlly under QMS, and then more researching leads, which are being developed by the USC investigators to give new capabilities not yet seen in commercial devices. Our IPG uses standard ball seal connectors, so other leads can be designed and used with the OpenNerve system. And we have the capability to connect thin film devices with standard lead bodies for translational testing, if you're interested. Because our devices are open source, everything we develop will be posted online for the community to use. So this includes design files such as PCBs, CAD models, and manufacturing documentation, as well as testing data will be posted on our GitHub page. We've previously updated our GitHub last June with our early prototypes, and we'll be doing another round of updates this June. So stay tuned to see all of the design files and the raw data from all the presentations earlier today. Animal data, as well as some other bench data, will also be released on the Spark portal for any investigators to use or look at as they need. And for other resources, center updates, and communication, you can check out our website at opennerve.org and follow us on LinkedIn. So I'll put all these links in the comments as well. So one of the most important parts of an open source project is choosing the right copyright license. And for our project, we're currently releasing files under the Creative Commons BY 4.0 license. This is a fairly permissive license, but it's not a copyleft license. And what that means is that you can use our files for anything you want, basically, whether it's academic or commercial, with the only requirement being that you give CARSS credit through a citation or other sort of mention. You can also build on top of all of these files, for example, through developing new firmware algorithms, modifying the PCB, or creating new electrode designs. and any modifications can be kept proprietary or even patented if that's relevant. So we really wanted our center to enable startups and entrepreneurs to commercialize new therapies. And so we believe this sort of licensing structure will be a good balance to allow proprietary development while still making sure it benefits the entire research community. Also, while it's not required in the license that you make anything you build open source, we are looking to continue growing CARSS and to continue adding collaborators. So, please think about contributing back to the project and making what you build open source as well. And if you want to talk to us about what that means, we'd love to chat. So I think it's important here to mention again that CARSS is really a consortium of 3 different organizations, those being MedAlly, Medipace, and of course USC. So each one of these organizations is contributing something different to CARSS, and therefore each organization is sort of specifically placed to help different collaborators with different parts of their project. So, for example, MedAlly, they're an incredibly great contract manufacturer with a ton of experience. They've developed and manufactured clinical level devices for DBS as well as for peripheral nerve stimulation. And for CARSS, they're contributing the IPG enclosure, the leads, as well as manufacturing capabilities. So for collaborators, they're available to help with both manufacturing devices, customizing any of the mechanical components, designing new electrodes or new lead bodies, or really anything else that a good contract manufacturer could do. Medipace for us is designing the PCBs, the electronics, as well as writing the firmware and software. And so for collaborators, they're able to do any sort of software customization, firmware and algorithm development, as well as electronics modification. And they also have a ton of experience in bioelectronic medicine. So they're a great resource to go to just for advice and consulting. And then at USC, we have 3 different investigators, each developing a different new sensor capability. We also have a lot of experience with thin film manufacturing. So please hit us up if you need any sort of capabilities of either manufacturing a thin film device or attaching a thin film device to a lead body for integration with our OpenNerve IPG. And then USC is also sort of coordinating the project and coordinating animal studies. So we're available to help with that as well. Now, on to the more practical sort of how could we help, or how can CARSS be relevant to your work? There's actually a lot of different options for working with CARSS, and it depends on the stage of R&D that you're in. For early exploratory research, for example, rodent research or acute tests in large animals, CARSS can provide bare PCBs and electrodes that just have bare wires for connection to your equipment, or whatever sensors that you need. And that can help you investigate mechanisms and get data quickly. For more developmental research, such as chronic large animal studies that are maybe not done under GLP, that are more investigating the efficacy of your therapy, we can also provide lower cost versions of our IPG and leads. This could include an IPG that uses, say, a primary cell instead of a rechargeable battery. It could be a different type of housing. For example, we could do a PEEK housing instead of a titanium housing. So if you don't need full hermeticity, we can provide that, as well as sort of avoiding some of the more potentially expensive components like ball seal devices. And then, of course, for the validation studies, the large GLP animal studies, and first in man, OpenNerve is specifically designed for that type of research. So we can provide all the documentation necessary to support an IDE application, and we can provide devices that can be used in animal studies and then immediately translated to human use without any sort of redesign. So working with CARSS earlier on in your research has some advantages when moving between these stages of development. For example, if you use CARSS PCBs for exploratory research or lower cost CARSS devices for your first chronic studies, you can validate a mechanism or a therapy and then bring it to the clinic using the exact same electronics, which saves the cost and time of developing a new system and would reduce the risk involved in transitioning between these stages of the research. You can also test custom algorithms, different feedback loops, different sensor inputs, or new electrode designs on small animals or acutely, and then take those into the large animal studies and the first in man studies without having to risk some sort of incompatibility between the clinical hardware and the new designs you've developed. In terms of cost and availability, right now we can only provide estimates, of course, as we're still in the design phase. So please reach out if you need a specific quote, and we'll figure that out for you. But in general, we've been providing bare PCB development boards to researchers that are interested in developing with the CARSS device for about $2,600, which is at cost for us. We're not making a profit on any of this. Right now, our first run of PCBs is all accounted for. Our next run should be ready in September. So let us know now, and we'll get you on the list for that. For the full OpenNerve IPGs, and these would be the hermetic titanium enclosures ready for sterilization and use in large animals or humans, the cost would be about $8K per IPG. Leads, depending on the lead design, would cost about $500 to $1,000 each. And for these, we need about 1 month of lead time. This is assuming that all of the components are easily procurable. Of course, delays happen. Supply chain stuff is still a little crazy, but for relatively low cost and short manufacturing time, we're able to get you this equipment to support your studies. And then speaking of funding, I wanted to give some examples of different funding mechanisms that our existing collaborators are using or have explored. So for exploratory research, R21, we've had people using these. There's several options for development, including this new R18, which is brain or heel specific at this point, as well as SBIRs and some funding through the NIH Blueprint MedTech program. And then for validation and first-in-human tests, our collaborators have gone through Blueprint MedTech, whether an optimizer or the sort of U44 or UG3, UH3 pathway. These are all, for all of these, we've supported you through anywhere from just giving a quote or giving letter support, all the way towards partnering as a co-investigator. And we're happy to support you in whatever way works best, either through one of these funding mechanisms or through whatever existing funding you have, or whatever funding mechanism you apply for. And this includes private VC funding as well. Like, we're more than happy to work with a startup or a company. So just to give a final example of how this might work, this is sort of an anonymized story of how we've worked with one research group. There was a professor that came to us that had a great idea, and they had rodent data that was very promising, but they didn't have a device for large animal testing, and they didn't really have a viable pathway to IDE. So they reached out to us, and we had a series of conversations and ended up really getting to know them, their therapy, and what they needed out of a system. It turns out that our existing IPG design would work really well for what they wanted, but they needed some variations in electrode design. And then, of course, they needed custom firmware that could implement their proprietary algorithm. So MediPace was able to support them with the firmware customization, and then MedAlly was able to build them the electrodes they needed. And we worked with them to support a NIH Blueprint MedTech application. Because they were working with us instead of developing the device separately, they were actually able to shave an entire year of development time off their Blueprint project plan, allowing them to hopefully get to IDE faster. So yeah, I hope you've enjoyed the webinar. If you have any follow-up questions or are interested in seeing if we can support your work, please don't hesitate to reach out either to our general CARSS email or directly to me. I'd be happy to chat, and I'd be happy to connect you to anybody that could help you with your work. Thanks, and we'll go into the discussion phase now.